This is lecture 3. Another way of looking at it is microbial lifestyles part 1. And the way I want you to think about that is as like an ecological niche. So what is that? Well, if I want to think about it in a simple way, I could think about it like, what does a wolf need from its environment? It's going to need some kind of food it can get and digest. So lettuce is not appropriate. Even though it can find lettuce, if it lives near a lettuce farm, it has to eat something different from that it can actually get. It needs a certain range of temperatures. A wolf cannot live at 100 degrees all the time, I assume. I don't know anything about wolves, but they're furry. What about a plant, though? A plant needs a certain amount of sunlight. If it gets too much, it gets burned. If it doesn't get enough, it gets bleached. Plants require certain type of soil that's porous to a certain degree, that either holds water or doesn't, and it needs water often or rarely. It definitely has a temperature range it needs. Some plants can survive freezing temperatures and others can't. Some can survive very hot temperatures and others can't. So any particular plant has to be able to survive where it is. It has to grow where it is, and it's either going to survive and thrive in that spot, or if it can't, it won't survive and thrive in that spot. And bacteria really are the same way. They need certain things out of their environment that allow them to reproduce. They need those things, and if they don't get them, they are going to be stuck in stationary phase, or they're going to die. Bacteria cannot move long distances. They cannot migrate from continent to continent. They are stuck where they are for all intents and purposes. And so if they don't have exactly the conditions they need, they won't grow, and they might not even survive. A single cell might not even survive. So we're going to look at really all aspects of that in these lectures. In particular, a lot of what we're going to think about are chemical and biochemical concepts. So that can be daunting to some students. And the whole idea here is for you to understand what a bacterium needs from its environment. And then we can apply this to fungi and protists as well. But it's important that you can start to see the world as bacteria see it. And so, like this slide tells you, some of the concepts we will look at are going to be fairly simple. Like we can talk about the temperatures bacteria need. There are a set of vocabulary words to describe different temperature ranges microorganisms might require, and you'll see that when we get to it. But in other situations, when we think about their metabolism, that gets more in depth, and you really will need to do a deep dive into these slides. Take your time and understand each slide. You will be able to, and once you've gone through them, you will have a great idea not only of how bacteria live, but you will understand your own cells a lot better. So I urge you to be patient with these and really dive in as much as you have to understand them. And we will all come out the other side a little bit more knowledgeable. We start with a very big question. Why are microorganisms small? And no one knows. We can't ask microorganisms why they are so small, and even if we could, they wouldn't know. It's like asking us, why do we have 2 legs? Why do we have 2 legs? Nobody knows. But biologists answer these questions through the lens of evolution. We say, well, in the understanding of modern science, any living thing is the way it is because evolution has pushed it to be that way, because organisms with those traits were successful in the past and they were able to reproduce and have offspring with those traits. And so we can think about small size of microorganisms through the lens of evolution. So we have kind of 3 different evolutionary hypotheses, and they don't disagree with each other. They could all be true, or none of them could be true. So the first one is the ratio of surface area to volume. And I'll explain this in the next slide. Another is diffusion of signaling molecules, and I will again explain this in the next slide. The last one is reproduction rates. So small sizes mean that it doesn't take long for bacterial cells to grow big enough to divide. And once they divide, they've gone through an entire generation, and they can mutate as they go through that process. And in the process of mutation, they can introduce genetic variability into a population that allows evolution to happen. It allows natural selection to happen. So the small sizes allow them to reproduce quickly, exploit new niches, and evolve quickly. So let's get into these first two that I didn't explain. The first one is the surface area to volume ratio, or the ratio of surface area to volume. And so we first talk about a cell's volume. So like how much cytoplasm is in a cell. And this correlates to how much a cell weighs, so its mass, but also its demand for nutrients. How many enzymes does it have inside itself? How quickly does it need to make new proteins? That all correlates to its size to some extent. And so if we think of a sphere, the volume of a sphere is, 4/3 pi r cubed, where r is the radius of the sphere, or the radius of a cell. So let's keep that in mind. That's how we calculate the volume of a sphere. And a sphere, and the volume roughly correlates to the cell's demand for nutrients. Then surface area. What does surface area correlate to? Well, remember that any kind of cell gets nutrients across its membrane. Nutrients have to cross the cell membrane to come in, and waste products have to cross the cell membrane to go out. And in many cases, the cell surface area is going to control how quickly that can happen. It's going to limit how many transport proteins there can be. It's going to limit how much passive diffusion there can be. So the surface area of a cell determines how quickly nutrients can enter and how quickly waste can exit. So we can calculate the surface area of a sphere at 4 pi r squared. And again, r is the radius of the sphere. So what happens if we make r bigger? So if the cells were bigger, what would happen? And that's what we should look at. Here we can get the ratio of surface area to volume. And again, we can think of it as surface area is the ability to transport chemicals, volume is the need to transport chemicals. So a happy cell has a lot of ability to do what it needs to do. So a happy cell is going to want to maximize its surface area and minimize its volume. And if we look at the ratio of surface area to volume from the formulas on the preceding slide, this is what we get, 3 over r. So as r increases, this ratio is going to get smaller and smaller. As this number gets bigger, this whole ratio gets smaller. So bigger cells have less ability to bring in nutrients and get rid of waste products. So that's that's surface area and volume. We just think that a very small cell has more efficient ability to move the molecules it needs across the membrane. And human cells, we don't really need that because we have a blood supply constantly keeping our cells fed. And other things like that. And our cells, most of our cells don't need to swim around and rapidly divide. Most of our cells are not in the process of doing anything like that. So they can be a little bit bigger. But most of our cells their radius would be like 10 times, 20 times bigger than a bacterial cell, not like 100 times or 1,000 times bigger. The last one is diffusion. So how quickly can like signaling molecules go from one end of the cell to the other? How quickly could a messenger RNA diffuse from one end of the cell to the other? And so here we could look at other physics stuff and say that the time required for a molecule to move a distance via diffusion is proportional to that distance squared. So if it takes a minute for a molecule to diffuse 1 centimeter, it would take 100 minutes to diffuse 10 centimeters. And that's what this is showing. If a cell gets 10 times bigger, it will take 100 times longer for molecules to diffuse across it. And so a bigger and bigger cell will need to solve a lot of problems related to getting materials from one part of the cell to the other. And yeah, this can be important for all of that. And this is why biologists are kind of amazed at things like motor neurons, which are enormous. And when I learned about motor neurons, my first question was, how do they get messenger RNA all the way to ribosomes at the distal end? How would proteins get to the distal end? How would anything get from one end to the other? Well, the answer is, if it's by diffusion, it won't. It could take, it's on the order of like a century for a molecule to diffuse a meter. So there must be other ways of making it happen, and our motor neurons certainly have those other ways. Okay, well that is enough for this first video. Next, we're going to start looking at the factors that really define microbial lifestyle. I delayed us a little bit to talk about why they are so small, but that should be the first question we answer in any microbiology course.